Hello students, in this session we are going to discuss about linkage mapping. Linkage mapping and we need to know about the definition of linkage first before going to the linkage mapping. I am Dr. Ashwath Narayan, lecturer in genetics. So here for the first, what is called as linkage here? Linkage is defined as the tendency or affinity of two or more genes remain together in original combination for number of generation in non-Mendelian fashion. So that means linkage tendency or affinity. Affinity means togetherness. Togetherness is the phenomenon where two, consider this is chromosome, in that A and B genes are there. A and B genes will be segregated to the one gamete because of shorter distance between them. So in crossing over, definitely the recombination will occur where the exchange of chromosomes, chromosome part will take place between non-sister chromatid of homologous pair, right? This chromosome is the common phenomenon, but linkage is rare, where you can observe that because of shorter distance between two genes, so they will be together for number of generation in original combination in non-Mendelian fashion. Non-Mendelian fashion means, in Mendel states that the genes will be segregated apart and enters into new gamete. But because of shorter distance, they will be together and they will not follow the independent assortment, that is called as non-Mendelian fashion. This mechanism is first discovered by T.H. Morgan, Thomas Hunt Morgan. He observed that in an organism called Drosophila, this is the model organism. Later on, many scientists discovered the linkage in maize, sweet pea and human beings. In Drosophila, body color and nature of being is linked together, located in the same chromosome. Sweet pea, the flower and size of the pollen is linked together because they are present in the same homologous chromosome. In maize, color and shape of the seed is linked together. And in human beings, some diseases and some traits are linked together. The common example is hemophilia and color blindness. Because of shorter distance between them, the person who is suffering from hemophilia will be suffer from color blindness. That is the linkage phenomenon. So how, you need to know the history of linkage here, how the person called as T.H. Morgan discovered the linkage. During his experimentation with Drosophila, he took a male wild type of Drosophila. That means wild type means eye color is red color and normal wing shape is there. So wild type of male he took and then crossed with a mutant type called as white eye and miniature wing. During his crossing, he got in F1 generation he got the wild type only, heterozygous wild type, I guess so. And later on, he need to test the parental combination. Again, he crossed that with the received parent. That means white eye and miniature will be received, right? And he do test cross with the received parent. So in test cross, definitely if tall is crossed with dwarf, in F1 heterozygous will be there. If you test cross with the received parent, the combination will be heterozygous as well as received trait. That means the parental combination is 50% and the parental combination is 50% and recombination combination is 50%. Like that, you'll get it. But in F2 generation, the maximum population will be the parental population. You can observe that after test crossing, he got to know that the 1541 Drosophila will be parental combination and lesser population will be recombination population. If you do the cross here, capital T, small t, and heterozygous and self cross, definitely the parental combination will be 50% together and recombination population is 50%, right? But in case of this cross, recombination fraction will be less than 50% and parental population is more than 50%. Around 75% will be parental combination and 25% will be the recombination combination. Primarily he thought of some other phenomenon like lethal gene interactions are there. Lethal gene interaction means either in homozygous dominant condition or hetero homozygous recessive condition that causes lethality and causes death of the individual, right? So this phenomenon he thought, but later on he concluded that this is not due to lethal gene interaction. This is due to linkage phenomenon. Linkage phenomenon, because of the more tendency towards these genes, they will be together in original combination. Because of that, if parents with same combination will be crossed, the triad will be inherit together, and they will be in more population as compared to recombination population. That is the history of linkage. Here you can observe the percentage of recombination concluded by the T.H. Morgan is around 36%. Is not at all 50%. Less than that, he got the recombination population, and he got around 66% of the parental combination. This is very rare phenomenon. This is rare phenomenon. But this is not the case here. This is due to the linkage. Linkage, because of that, this original combination will be retained. Because of that, parental combination is more as compared to recombinants. Based on this information, Morgan postulated a theory called as, or a Morgan linkage analysis will be put forward. According to this theory, this is also called as the principle of linkage. The theory states that, or Morgan's linkage analysis states that chromosomes contains many genes. We already know that. No, this is the chromosome, and it contains many coding part. It contains heterochromatin, in euchromatin. In euchromatin, you will get genes, and genes will express something, right? That is, chromosomes contains many genes, and genes are arranged in linear fashion. We know that, in karyotyping, so the heterochromatin band, euchromatin banding will be there, and euchromatin banding will be in serial order 1, 2, 3, 4 like this. So we already know the nomenclature of karyotype, the ideogram. In that, we can compare the numbers here, so to detect the mutations and aberrations here. So in that, genes are arranged in linear fashion, and some genes which is present in chromosomes are linked. So that is the phenomenon here. That means, because of shorter distance between them, consider A and B genes are there, they are linked together as compared to the combination between A and C. A and C segregate to different offsprings because of longer distance between them. So they will be separately independent assorted to different gametes here because of longer distance. Some genes, because of short distance, link together. They are called as linked genes here. Linked genes will be inherited together from parent to offsprings without segregation. That's a very crucial point for the linkages here. The linkage is rare phenomenon as compared to crossing over. We know that crossing over is 99%, the genes will be crossing over, and recombination frequency will be more, and linkage will be very rare phenomenon. And strength of the linkage depends upon the distance between the linked genes. Here, you can see here, this is chromosome, and A and B and C is there. A and B will have 5 centimorgan distance, centimorgan, and A and C will have 15 centimorgan distance. In that case, you can see AB combination will comes under complete linkage, strong linkage, as compared to AC combination. Why? Because AB will have shorter distance of 5 centimorgan, whereas AC combination will have the distance between them is 15 centimorgan. Because of that, it comes under incomplete linkage, right? So the strength of the linkage depends upon the distance between them. Shorter the distance, more linkage. Longer the distance, less linkage will be there. And here one more phenomenon you need to remember here, Mendel laws of independent assortment is not universal, because linked genes will not participate in crossing over because of the short distance. These two genes will not participate in crossing over because of short distance. Because of that, we can state that Mendel law of independent assortment is not universal. And as I said earlier, linked genes do not show independent assortment, but in weaker linkages, like complete linkage and incomplete linkage, over the period of time, these two linked genes will be segregated. And one more phenomenon, one more type of linkage we can identify. You can see here, this is homologous pair, right? This is homologous pair is there. In that some genes are there, some genes are there, in that A, B is here, and then small a, small b is there, right? If dominant genes which is present in the homologous pair is called as cis arrangement. If heterozygous type of combinations are there, instead of that capital A small a is there, and here capital A small b is there, and small a capital B is there. This is rare combination. This is called as trans arrangement. We're going to discuss that in next successive slides here. And all linked genes in homologous chromosome constitute a group called as linkage group. So the linkage group is directly proportional to the number of chromosomes they attain. In that case, you can see the number of linked groups in an organism, that is directly proportional to number of pairs of chromosomes. In Drosophila, 4 linked group is there, 4 pairs of chromosomes is there. That means because of 4 pairs of chromosomes, Drosophila has 4 linked groups. In pea plant, 7 pairs of chromosomes are there. We already know that in Mendel experiment. And in human beings, will have 23 pairs and 23 linked groups are there. And then linkage is 2 types, complete linkage and incomplete linkage. As I said earlier, complete linkage do not break because of the shortest distance between them, and incomplete linkage soon segregate in next generations because of weaker strength, because of short and longer distance between them. Example for complete linkage is the wing shape and eye color in male Drosophila, wing shape as well as eye color in female Drosophila is incomplete linkage. So types of linkage here, based on the strength of the linkage, we can categorize them into complete linkage and incomplete linkage. Here, as I said earlier, because of the shortest distance between them, 2 genes never segregate and never go for Mendel independent assortment. Example for that, body color and shape of the wing is of male Drosophila. Remember, this is easy to remember. Male Drosophila, body color and shape wing is completely linkage, and in incomplete linkage, because of some longer distance, occasionally they will separate and leads to formation of new recombination. The recombination product will be emerges out, but those recombinations will be lesser number as compared to parental combination. Parental combination, you can observe that 52, 60, 75% will be parental combination, and only 25% will be the recombination, new recombinations will be there, even though they are there in, they will participate in crossing over. In normal crossing over, 50% will be, sum of parental combination will be 50, and remaining thing will be recombination population here. Based on the genes involved, as I said earlier, coupling and repulsion will be there. 2 dominant genes in one homologous pair, 2 receive genes in other opposite homologous pair is called as coupling phase or coupling linkage, where same chromosome and will receive dominant allele, and other chromosome will receive opposite homologous chromosome receive allele is called as cis arrangement or coupling phase, where heterozygous conditions will be there in both homologous pair. That means TR, capital T small r or small t capital R. This is heterozygous combination in both homologous chromosomes are there. This is called as trans arrangement and also called as repulsion phase. Based on the chromosomes involved, that means it may be autosomes and allosomes. You already know that this is somatic chromosomes that mainly gives our phenotypic trait, and this is sex chromosome or allosomes, where there also we can find some linkages here. Hemophilia and color blindness is X-linked recessive. Then you can observe that X-linked means definitely the genes which is defected with respect to hemophilia and color blindness are present on the X chromosome. In calculation of the linkage group, linkage related problems, some formulas will emerge out. The first formula is the recombination frequency. Recombination frequency is equal to total number of sum of recombinants total sum of recombinants divided by total number of offsprings. Remember, total number of offsprings. The recombination frequency is important to determine the linkage groups here, a linkage phenomenon. In that, recombination frequency is equal to the sum of recombinants divided by total number of offsprings here. That is the formula for recombination frequency. As a next, one more phenomenon is called as coefficient of coincidence. What is coefficient of coincidence? Is the measurement of, statistical measurement or calculative measurement of the interference in crossing over. Take example here, this gene, this is one gene, this is other gene. They tends to crossing over during meiosis, right? During meiosis, they tends to crossing over and recombination type of chromosomes are formed. In that, some mechanism is there that will interfere in chromosomal crossing over. So those things are called as interference, and to measure that interference, we need coefficient of coincidence. This formula is also very important. Why? Because you can see if A, B genes are B genes are there, A, B, C genes are there. In that, we need to find the pattern of arrangement of chromosomes. We need to find which crossing over is favors. In that, two crossing over is called as double crossing over. Single crossing also may also occurs here, and different recombination patterns will be there. To calculate that, we need to calculate coefficient of coincidence here, right? In that, how to calculate that is, coefficient of coincidence is equal to sum of double crossing over product, divided by, distance, assume that, A, B, C genes are there, and small a, small b, small c genes are there. In that, if assume that this is the spot of crossing over and this is the spot of crossing over. If you measure the distance between A and B with some formulas, it is also equal to A and small a and small b. Why? Because they are they are present in homologous pair, and if the spot of A is exactly where small a also present, right? Because of that, if the only assume that these are two spots here, in that this is this first recombination frequency. Assume that only this this crossing over only happens here in this spot. Then recombination occurs here. I will rewrite once again. A, B, C is there, small a, X, Y, Z. Assume that X, Y, Z is there. If assume that only this in this spot the recombination occurs, the first recombination product is equal to A, Y, Z. Y, Z is the single crossing over product in the spot of A and B or X and Y. Assume that one more product is X, B, C. This is other crossing over product with respect to distance between A, B or X and Y. Assume that in the same combination A, B, C is there, X, Y, Z is there. In that, instead of here, the crossing over happens here. If you calculate, if you know the recombination A, B, Z will be the one recombination, and other recombination is X, Y, C is other recombination. This is the single crossing over product with respect to BC or YZ. This is single crossing over product with respect to AB or XY. Assume that this is the combination, the previous combination. So simultaneously two crossing over happens. Then what is the combination? This is called double crossing over. First crossing over happens here, it will joins to Y, and again one more crossing over happens and it will joins here. The double crossing over product is equal to A, Y, C, and then X, B, Z. Understand? The middle gene will be segregated independently to different chromosome. You can see middle genes will be segregated independently, but extreme genes will together segregated to one chromosome. And see, AC will be segregated to one chromosome, leaving back the middle gene B here. To calculate the parental combination, we need to find double crossing over product first. So we'll discuss that in solving problems here. First, we need to know what is coefficient of coincidence. It is a measure of interference which interfere with the crossing over. So this is the formula you need to remember. Coefficient of coincidence is equal to sum of double crossing over product divided by distance between AB. So assume that this is ABC is there. we need to find first distance between AB and then distance between BC. So the whatever distance is there, that should be in Morgan distance, not in centimorgan. Morgan distance will be there. The distance between AB and multiply by distance between BC, and that into total number of offsprings is the formula with with respect to crossing quotient of coincidence. One more phenomenon that mainly happens in linkage is interference. We're going to discuss that. Interference is the impediment that happens during crossing over. Impediments that happens in crossing over. Interference is equal to 1 minus coefficient of coincidence. To calculate interference, we need to minus that with the number here. Okay, that is the formula for interference. So in the next one is genetic mapping. You already know that what is genetic mapping. In that to map the distance between each gene is called as genetic mapping on the chromosome, which is it's a systematic method to measure the distance between the coding part of the gene, right? In that the formula is crucial. We already discussed about it. Recombination frequency is equal to sum of recombinants divided by total number of offsprings. If you want that in percentage, definitely you need to multiply by 100. That is the recombination frequency percentage, right? Next, we will move on to problems here. The first problem is gene ABC is located on the same chromosome, and it is not necessary in the same order. You need to mention that not necessary in the same order, that is arranged on the chromosome. The recombination frequency is given in the below tabulation, and find out the order of the genes on the chromosome. It is easy. Why? Because recombination frequency is directly proportional to the crossing over, right? That means if recombination frequency is more, then distance between them, the genes, will be more. If more recombination frequency is there, they are more likely to go for crossing over. So because of that, more recombination frequency, you need to find to find the extreme genes, you need to find the maximum number of recombination frequency. Among those, BC will have a maximum recombination frequency. That is the key here. BC should write that in extreme. BC will be the extreme genes, and later on we need to move with the next percentage, next excessive percentage, lesser percentage. BD, that means BD will be 40%, B and D distance will be further and lesser than the BC. Because of that, we will write D here, right? So BC will have 45%, BD will have 40%. And one more combination is there, AB. AB is there, definitely A will be there here. This is the combination of genes here. AB will have 30% of recombination frequency, and AD will be there, that is less than 25. So this is the combination of the genes on the chromosome B, A, D, C, or C, D, A, B. So this is vice versa. So like this or like this, the combination will be there. This is the combination of the arrangement of genes on the chromosome. Again, one more problem we're going to discuss, that is gene ABC is located on the same chromosome, and mention not necessary in the same order. Not necessary in the same order. Same order chromosomes are arranged, and recombination frequency is given, and we need to find the order of genes here. As I said earlier, you need to find the more recombination frequency products here. So C and D will be extreme here, 48%, and then next one is lesser than that is 25%. AC, that means A will be here, and then AD will be there. AD will have 23 here, 23 here, 25 here, and then BC will be there. BC will have lesser distance B here, and it is 15%. Like that, BA will be there, and then 10%. This is the combination of order of genes and the chromosome CB, AD, or DA, BC. So like this or like this. So this is the combination of genes here. This is the order of genes, sorry. Next, we will want to move on to other problems here. In Drosophila, flies red eye is dominant over white eye. Normal wing is dominant over vestigial wing. If these genes are crossed, red eye and that means wild type is linked together, and then vestigial and white eye is linked together. A test cross is conducted. Following offsprings are produced. And what is the most percentage of recombination frequency for that? You already know that linkage, in linkages, the parental combination will be more. Parental combination will be more as compared to recombination population. So in that easily we can find the parental combination 368 and 336 will be the parental combination, and these are the recombination products here. We need to sum that. Recombination frequency is equal to sum of the recombination frequency, recombination population, or recombination population divided by total number of offsprings here. We need to sum up this one first. We need to sum up this population, and we will sum up first. So first, 368 plus 56 plus 336 plus 40, that will give you 800, right? So here 56 will be the first recombination product here, and 40 divided by 800, and we'll sum up 56 plus 40, then it will the value is 96 divided by 800, then 96 divided by 800. So that is equal to the recombination frequency is 0.12. Recombination frequency is equal to 0.12. You want to multiply and one percentage of that into 100. If you do that, definitely 12% will be the recombination frequency, right? The percentage, the most percentage of recombination frequency is equal to 12%. Why? Because they asked in percentage here. Otherwise, recombination frequency will be 0.28, 12. Sorry. Next, we'll move on to next one more problem here. Same thing. Gene DEFG is located on same chromosome, not necessarily in the same order. Distance between them are given in centimorgans. So previously, what they given, either they will give recombination frequency, either they ask to calculate recombination frequency, but here different parameters are given. That is, map distance is given. Same thing, map distance and recombination frequency is same. Map distance means directly they are giving distance between them. In that, extreme distance is GF. That means extreme gene should be GF. It contains, it is 22 centimorgan distance here. Next, the lesser than that is 15 here. That means EF. F is there, E is there. We need to write that at the corner of G here. And then 11 is there, GD. And then D will write in opposite corner. And then lesser than that, GE is there. GE is 7 centimorgan. DE is there. DE is 4 centimorgan. DF is there. No DF, right? Okay. And then GD is given. GD is equal to 11 centimorgan. GEDF or FDEG will be the pattern of arrangement of chromosome, order of arrangement of chromosome here. Next, we will move on to again one more problem here. In fly, smooth wing is dominant over wrinkled wing, and red body is dominant over yellow body. So the mating between the heterozygous with receive parent, it is heterozygous with the receive parent, following offsprings are produced. What is the most percentage of recombination frequency? So you need already in previous one problem we discussed about it. The maximum population will be the parental combination, and then the lesser population will be the recombination population here. So in that, the formula is equal to recombination frequency. Recombination frequency is equal to sum of the recombinants divided by the total population. So sum is 162 divided by 158 plus 162, 158, 778 plus 785. We'll sum up first here. 162 plus 158, so 320 plus 778 plus 785. So the number is 1883. Right? If you divide that 320 divided by 1883, the recombination frequency is will be 0.16. If you multiply by 100, it will give the percentage 16% will be the recombination frequency. This is the answer. Next, we move on to one more problem here with respect to linkage mapping. The question is given like this: in trihybrid cross, the between capital A, small b, capital C, capital A, small b, capital C, cross with small a, capital B, small c. Not necessarily in the same order. So this combination will be not necessarily in the same order. It may be BAC will be there, CAB will be there. We don't know. The following offsprings are produced. I think 1, 2, 3, 4, 5, 8 combinations is given. In that, the question is find out the distance between two extreme genes. So in that normal, if the 3 genes are there, based on that, based on the recombination and non-recombinant products, we can easily calculate that. But here many recombinants are given here. So how to find out that? We're going to discuss that in this session here. In that, find out the distance between two extreme genes, and next question is find out coefficient of coincidence, and third question is calculate the interference. In that, we need to know about, this is trihybrid cross, right? Because of that, we need to find what and all the which is present here, which is belong to which one. In that case, we need to find first. We need to find first related to which one is which and which combination is which, right? So because of that, we need to first go for finding out the parental combination and double crossing over parental combination. Assume that A, B, C is there, X, Y, Z is there. If the crossing over happens in this part, this is called a single crossing over product. Here it will gives out the combination like this: A, Y, Z, and then X, remaining thing X, B, C. This is the single crossing over product with respect to AB or XY. Assume that in the same combination ABC, XYZ is there. Instead of here, so here the crossing over happens. The combination will be ABZ and XYZ. ABZ and XYC will be the single crossing over product with respect to BC as well as YZ. One more combination will be there, ABC, XYZ is there. In that, instead of single crossing over, simultaneously two crossing over happens. In that condition, the product will be double crossing over products. Here, A first, crossing over happens with Y, and again crossing over happens with C here. That the product will be A, Y, C, and then X, B, Z. It is plus double crossing over products here. So in that double crossing over products, you need to find out, we need to know that with double crossing over product, we can easily find out the parental combination. Why? Because you can see, if you find out the extreme genes in one genotype will be inherit together. You can see A, C is there. They will together inherit to the next gamete here. But the middle gene will be segregated independently to the other gamete here. So here extreme genes will be remain together if you find out these extreme genes, and if independently one middle genes are segregated, definitely the pattern of arrangement of these chromosomes will be identified through double crossing over product. How to find out that? We'll do that now in this session here. We'll rub up this first and then we'll move on to find the parental combination and double crossing over products, and then from the double crossing over product we'll find out the arrangement, pattern of arrangement of the parental combination, and then we'll find out the distance between extreme genes here. So here, as as familiar, the parental combination will be more. Because of that, we will call them as parent 1 and parent 2 easily. And one more easy way to find out double crossing over product is double crossing over product will be rare. Because of that, the whatever the population is very meager. So this is double crossing over product with respect to double crossing over product 1 and double crossing over product 2. And whatever remaining things are there, so that will be identified after the combination. So as I said, as I mentioned in this genotype cross, so it is not necessary in the same order that is arranged. That means, to confuse people, it may be BAC is given or ABC is given. How to find out that arrangement of chromosome is by double crossing over product. I will write down two double crossing over product here: capital A, capital B, small c, small a, small b, capital C is given. This is double crossing over product. We will compare with one parent here. So this is one parent, capital A, small b, capital C is given. In that, we can easily find out the extreme genes and middle gene here. As I said earlier, extreme genes will be segregated in, segregated together, where the middle genes will be segregated independently to different gamete. As I said earlier, this is small b and capital C is there. Find out small b and capital C here. Small b is here and capital C is here, and because of that, the combination extreme genes will be small b and capital C, right? And to find out the middle gene, you can see capital A gene is independently segregated without any other genotypes here. That means definitely A will be in the middle. This is the common arrangement, pattern of arrangement of chromosomes, pattern of arrangement of genes on the chromosome. To recheck that, again we'll compare that with one more double crossing. We'll write with one more parent here, capital A. We'll write double crossing over product once again here, ABC, small a, small b, capital C. So this is two double crossing over product we written, and compare with one more parent here, small a, capital B, and small c is given here. Find out the extreme genes here. We will be segregated together here. As I said earlier, B and small c is segregated together, right? Definitely capital B and small b will be segregated together. Because of that, they are extreme genes. And then small a, you can observe that they segregated independently to one genotype or one gamete here. So here the combination, the parental combination, you can replace this with small b, capital A, capital C, and replace this with capital B, small a, small c. So this is the parental arrangement of chromosome, here, and parental arrangement of chromosomes here. We can easily find out that, the other combinations here. Okay, as quickly as possible we'll find out the extreme, already we find out the extreme genes, and we'll go on to calculate the distance between them. Okay, and yes, we already know the pattern of arrangement of the parental genes on the chromosome. Small b, capital A, capital C, capital B, small a, small c will be the two parental combination. To find out, we'll find out first double crossing over product with respect to BA or BA, this BA or this BA. Assume that this crossing over happens, it will gives rise to this combination. So first combination is small b, small a, small c, and this combination is capital B, capital A, capital C, this combination, right? This is single crossing over product with respect to BA, or whatever things are there. Next, we'll write once again the parental combination, small b, capital A, capital C, capital B, small a, small c, and instead of that, this crossing over happens here. This is single crossing over product with respect to AC. Why? Because this is distance between AC, right? So in that, pardon me, the combination will be BAC, small b, capital A, small c, and this is the combination BAC here, capital B, small a, capital C. If you want to write one more time, I will write it. Small b, capital A, small c, capital B, small a, small c is there. Instead, in this location, recombination is happened, and that will gives rise to this combination, and this will give rise to this combination, small b, capital A, small c, and pardon me, capital B, small a, capital C is given. This is the single crossing over product with respect to AC. Why? Because this is the combination BAC is there. First, we'll find out distance between BA and distance between AC, and we merge together and find out the products here. Understand? We calculated the single crossing over products here. We find out the single crossing over product BA and single crossing over product AC here. Find out which one is which here. Small b, small a, small c. That means small b, small a, small c is there. This is this label it single crossing over product with respect to BA. One more combination, capital B, capital A, capital C, capital B, capital A, capital C. This is single crossing over product BA. Okay. Remaining two will be double cross, the single crossing over product with the AC. We'll check it out. First, check it whether correct or not. So here, small b, capital A, small c, small b, capital A, capital C, small c. So this is single crossing over product with respect to AC. One more combination, capital B, small a, capital C. So capital B is there, capital C is there, small a is there, single crossing over product AC. Understand? So we already find out the pattern of arrangement of genes on the chromosome. Then we'll find out the distance between BA first, followed by AC. Then we'll sum up BA and AC to calculate the distance between the B and C here, because B and C are two extreme genes which is present here. So in that, we need to proceed with calculating the distance between BA or small b or capital A, whatever, small b, capital A is equal to sum of single crossing over product with respect to BA plus sum of double crossing over product divided by the total number of population is the formula. So in that, sum of single crossing over product is equal to this single crossing over product which is given here is BA and this one, this combination 46 and 38 is given. So in that case, 46 plus 38 plus sum of double crossing over, instead of that 4 plus 2 is the combination, total will be 1000, right? 46 46 plus 38 plus 4 plus 2, and in that case, you will get 46 plus 38. Is that, we're going to calculate the total individual cell 46 plus 38 plus 4 plus 2, 90 is that 90 divided by 1000, and you'll get 0.09, right? 0.09 is the distance between B and capital A. This is in Morgan, and then this is also important here. This value is also important, and then we can convert that into centimorgan. Distance between B and A is equal to in centimole 0.09 into 100, that will be converted into centimorgan here. This is 9 centimorgan distance between BA. Next, same, we need to calculate the distance between AC. Distance between AC, small, capital A, capital C, yeah, capital A, capital C. Distance between AC is equal to sum of, so single crossing over product with respect to AC plus sum of double crossing over product divided by total. So in that case, you can see sum of single crossing over product with respect to AC is 1 to 71 plus 63, 71 plus 63 plus 4 plus 2, 4 plus 2 divided by total is 1000. That is equal to the total 71 plus 63 plus 6 or 4 plus 2, 140, and 140 divided by 1000. So that comes under 0.14 Morgan. If you want to convert that into centimorgan, 0.114 thousand, 0.14 into 1000, 14 centimorgan. So we calculated distance between BA, correct? We calculated the distance between BA, right, and distance between AC, right? So this is distance between AC, is 14 centimorgan, right? And this value is also important. So then the combination distance between extreme genes here, B, C, right, small b, capital C is equal to distance between BA plus distance between capital A, capital C. So we already calculated distance between BA 9 centimorgan. M should be capital, and distance between AC is 14 centimorgan. So total distance is equal to 23 centimorgan. That is the distance between two extreme genes. Distance between first question answer is equal to distance between extreme genes is equal to distance between small b and capital C. That is equal to the combination of small b capital A plus capital A capital C. Distance between that we already calculated that, and then if you sum up that in centimorgan, the distance between that will be calculated. The first part A question answer is over. Next we move on to B question, what is called as coefficient of coincidence. Coefficient of coincidence is equal, the formula is all double crossing over product, divided by distance between A, distance between BC, small b, BA, sorry, distance between BA in Morgan, distance between AC in Morgan into total. We already know that all double crossing over product means 4 is one double crossing over, 2 is other double crossing over product, and distance between BA in Morgan. So Morgan distance is equal to 0.14 with respect to small b capital A, and distance between sorry, not 0.14, distance between BA is 0.09, and one more distance is distance between AC is 0.14. Distance between AC is equal to 0.14. 0.09, this is 0.09, 0.14, and total will be 1000. So calculate that. 4 plus 2 is 6 divided by, we calculate distance 0.09 into 0.14 into 1000. So the value is 12.2, and quotient of coincidence is equal to 6 divided by 12.6, that is equal to 0.14. 0.47 is coefficient of coincidence. The second sub question answer is 0.476, right? Next, this quotient of coincidence is unitless. You need to remember this is unitless. There is no unit at all. And the next question is the what is that? Interference. Interference, you already know that interference is the interfering mechanism that works in crossing over. That is interference. The formula is 1 minus the quotient of coincidence. You already know that 1 minus quotient of coincidence is calculated 0.476, and if you calculate that, the value will be 0.524. This is the interference. So if interference is more, definitely the linkage will be lesser. Interference is more and linkage will be lesser. This is also unitless. Understand? We calculated the distance between extreme genes, coefficient of coincidence and interference. If you are still in confusion, we'll move on to same problem move on to the same type of problem in next slides here. Same question here in trihybrid cross, the following offsprings are produced. So it's a different types of symbols are given. Instead of ABC, capital ABC, they have given a different symbol here. We'll convert that into symbols here. Plus 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 is given. Assume that plus 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 will be capital here, capital A, capital B, capital C. And then this is small a, small b, small c. Whatever the combination is there, you believe that this is not necessary in the same order. Remember, not necessary in the same order. Without comparing with double crossing over product, whichever double crossing smaller number is there, we need to find out first, right? After that only we can find out the pattern of arrangement of the parental combination, right? Not necessary in the same order. This parental combination, we already know extreme maximum number will be parental combination, parent 1 and parent 2. And then so this is capital A, capital B, capital C. This is small a, small b, small c. Next one is small b, capital A, capital C will be there. This combination will be 64, and this combination will write it plus is B. Assume that B and small a and capital C, right? Assume that this is capital B, small a and small c, and this one is capital B, capital A, small c, and this one small b, capital A, small c, and this one small b, small a, capital C, right? In that, this one we find out the parent numbers here, and which one is least numbers are there among this population will be double crossing over product. You can see 10 and 15 is double crossing over product because they are least here, double crossing over product. Then we'll write down the combination here. Don't write it, it will be confusing. Instead of that, write this one, double crossing over product B, capital B, small a, capital C. This is one double crossing over product, and one more double crossing over product is this one, small b, capital A, small c, right? These two are double crossing over product. We'll compare with the one parent here. First parent is capital A, capital B, capital C. Same thing, you will write it, capital B, capital B, small a, capital C, small b, capital A, small c. This is two double crossing over product. I know whatever I abbreviated, I written that in this combination, right? Instead of plus plus, it will be confusing. I have written that is in replacing with capital letters here, and comparing with one more parent combination, small a, small b, small c. In that, how to find out the pattern of arrangement of chromosome is, as I said earlier, two extreme genes will segregate together and middle genes will be separately segregate to one of the gamete. Find out that, you can see B and C will segregate together. Definitely BC will be extreme and A independently segregated, right? Independently segregated to one of the double crossing over product, that is A. Again, you need to reassure that B and this is small c will be segregate together, together, that means they are extreme, small c, and A is independently segregated and small a. This is the combination of the parent. That means B, capital B, capital A, capital C, small b, small a, small c. This is the parental combination. By this, we can find out the single crossing over product here. Single crossing over product means, you can see, if recombination, assume that this is the parental pattern of arrangement and this single crossing over product happens between B and A. That means if that will happen, so you can see the combination will be this pattern. This is capital B, small a, small c, and this pattern will be like this: small b, capital A, capital C. This is single crossing over product with respect to BA. Next, we'll move on to find out this crossing over. That means, I will write it one more time: BAC, small BAC here. Instead of this position crossing over happens. The combination will be like this: capital B, capital A, small c, and small b, small a, capital C. So this is single crossing over product with respect to AC. Label it here. Find out capital B, small a, small c, capital B, small a, small c. This is single crossing over product with respect to BA. Next one is small b, capital A, capital C. Small b, capital A, capital C. This is single crossing over product with respect to BA. Next one, we'll write down the other single crossing over product: capital B, capital A, small c, capital B, capital A, small c. This is 100. This is single crossing over product with respect to AC. And remaining thing is 95. We need to check that the genotype is correct or not. Small b, small a, capital C, small b, small a, capital C. This is 95. This is single crossing over product with respect to AC. We already find out the pattern of arrangement and then single crossing over product. Then we will calculate the distance between two extreme genes here. I will wrap it up first. We find out the single crossing over products with respect to B and AC. Why? Because we already know that B, AC is extreme genes here. Small b, small a, small c is extreme genes here. And BC is extreme genes. A should be in the middle. And to find out the distance, first we need to calculate the distance between BA. We already know the formula: distance between BA is equal to sum of single crossing over product with respect to BA plus sum of double crossing over product divided by total. Right? Sum of single crossing over product with respect to BA is equal to, here BA is here, BA is here, 64 plus 66 plus sum of double crossing over product 10 plus 15 divided by total. The total will be 1000. So we calculated the total will be 1000. So the 1000 will be there. If you sum up that, definitely the answer will be I think 0.155 will be there. This is Morgan, and if you convert that, you multiply that with 100, it will be converted into centimorgan. 15.5 centimorgan distance. This is the distance between BA. Right? If one more combination is distance between AC we need to find out. Distance between AC is equal to same formula, sum of the single crossing over product with respect to AC plus sum of double crossing over products divided by total. If you calculate, sum of single crossing over product with AC here, 100 and 95 is there. 100 plus 95 plus here 10 and 15 will be there. I will write directly 25 divided by 1000. So the value will be 0.22 Morgan will be there. If you multiply that to 2 into 100, it will give 22 centimorgan distance. So next we will move on to calculating the distance between extreme genes. We already know the combination BAC will be the combination. First, distance between BC is equal to distance between BA plus distance between AC. So here distance between BA is calculated. That is, you can mention that in centimorgan. So all of the things should be in centimorgan. So because of that, 15.5 will be the distance between BA, and then 22 centimorgan will be the distance between AC. Then if you sum up that, the total value will be 37.5 centimorgan. This is the first A question answer, the distance between extreme genes, right? The second one is quotient of coincidence. You already know the formula, quotient of coincidence B. So here B, the quotient of coincidence is equal to sum of double crossing over product divided by sum of double crossing over product and the distance between BA in Morgan, distance between AC in Morgan into the total number of offsprings. That is equal to sum of double crossing over product means here 10 plus 15 divided by distance between BA in Morgan. The Morgan distance is 0.155 into distance between AC is 0.22 into 1000. If you calculate the value, the quotient of coincidence will be 0.733 unitless. Quotient of coincidence is unitless. Then you can easily calculate the third quotient, that is interference. Interference is equal to 1 minus quotient of coincidence 1 minus 0.733, that is equal to 1 minus 0.733 is equal to 0. If you minus 1 minus 0.733 is equal to 0.267, and interference is lesser. Definitely the linkage will be more. This is the conclusion you can draw, that this is the actual calculation part here with respect to distance between extreme genes, quotient of coincidence and interference. In examination, this is very important. Even they will ask in CSR examination, you have to calculate everything orally. So that's for this video. That's up. Thank you. Thanks for your support.